And look at the beginning part. That's pretty much identical. It's the same. And furthermore, when they looked closely at the protease gene, they found that the first part of that sequence codes for threonine, alanine, alanine, which tells you kind of quickly where that gene came from. It's a broken enzyme. There was an error in replication that destroyed the function of one copy of this enzyme and produced a gene that had the same sequence and then just had this threonine, alanine, alanine thing sticking off there. And it's all produced, this gene just produced threonine, alanine, alanine, nothing else. So that's where it came from. It's a broken gene. And to confirm that, the other cool thing about the antifreeze protein is to figure out how the antifreeze protein is produced. It's produced in the pancreas, and it's squirted into the intestine, where it's then restored by the intestine in the bloodstream. A very cumbersome way to do it, kind of a silly way to do it, but that makes sense. And what you've done is taken a pancreatic enzyme and modified it. The other cool thing, so how did that gene get in the first place? Well, one common process, you get duplications of genes. And so there was an ancient duplication that produced a copy of the enzyme, and then mutation could modify the copy, leaving the original intact so it could still digest stuff. And this kind of duplication event occurs all the time. And one other cool thing they discovered in this fish genome is this third one right here. They found a third copy, a partial copy that was made later in the process of producing, evolving the enzyme. And that is, it's all broken up, and what you've got instead is little fragments of the orange bits of the pancreatic enzyme. You see those over there. And you find smaller chunks of the threonine, alanine, alanine sequence. So it's like an intermediate step. It's a transitional molecular fossil inside the fish genome. Now, how cool is that? So we're finding lots of examples like this where you can actually trace through the genome and sort of figure out where these enzymes came from. They came from modification of pyrite enzyme. And sometimes you can get very radical sorts of differences. And here's another example. This was discovered in Japan, where apparently industry is kind of sloppy. And there's lots of plants that make nylon. And they sort of squirt wastewater all over the place. So you go to these factories that produce nylon, and you can find these glowing green ponds all over, full of precursors to nylon that were just sort of leaked out of the plant. You would not want to drink these. This would be awful. You know, horrible chemical smells, all this kind of stuff. So I find these wastewater ponds that are full of the byproducts of nylon production. And again, I mentioned to you, nylon is a recent human invention. We made this, we invented this, we devised chemical ways to produce it. It did not exist before human beings discovered it. And what they find in these ponds are bacteria that are eating nylon. Really cool. They have adapted to all this waste product, and they've now got an enzyme that can break down nylon and use that to generate energy. This is something new. Bacteria did not have this before. There was no nylon before. When they sequenced these things, what they found is bacteria have these little small pieces of DNA called plasmids. So this is an example of plasmid. It's a little circular piece of DNA. And they sequenced and they could find the enzyme on there. So the little black rectangles, those are the enzyme. The white ones are other enzymes that are found in other bacteria. So they found these, and like with the ice fish, they found different intermediates. So some of those black enzymes, they're really good at breaking down nylon. Others are not so good. So they can actually see, again, selection caught in the act. They can see some of the enzymes that work really well, some that don't. Here's the other thing. They figured out where it came from. This is the result of a frame shift mutation. So I'm going to explain a frame shift mutation. You know, the DNA is encoded in a triplet code. You read things in threes, and that's how you get amino acids sequenced from nucleotides. So everything's read off in threes, and you can imagine an imaginary language where every word was 3 letters long. So for instance, the cat ate the rat, okay? That's like DNA. A frame shift mutation involves, for instance, a deletion of one of those letters. So if you delete the E in the, everything frame shifts a little bit, and now what you have is that sequence, THC, ATA, DET, HER, ET, and you would say that's nonsense. That doesn't say anything. The way this nyelinase enzyme was produced is that there was a frame shift mutation that produced a random sequence of nucleotides and DNA. And when it's transcribed into a protein, that protein had enzymatic activity, just by chance. So here's a case where we have the novel origination of an active enzyme, just by chance. Initially, it was not a very good enzyme. It took selection to make it much better. Okay, let me give you one more example. And I'm not going to go into a lot of detail here, although this is a beautiful story. You should look it up. Look up Joe Thornton. I think this was published in Science a couple of years ago. And it's a beautiful example of working out the details of the origin of particular enzymes in us. That what we have in us is a couple of hormones and receptors that do different functions. One of them is a hormone called cortisol. You've heard of this? It's a fairly common one. If you've got inflammation, your doctor may prescribe it for you because it's a hormone that signals to the cells to calm down, stop getting so worked up, let the inflammation die down. So it's an important signal that way. There's another one called aldosterone, which is a hormone involved in salt balance. So you're probably not familiar with this one because this is one that, for instance, acts on your kidneys and tells your kidneys to take up or not take up as much salt from the blood. So it helps you maintain your salt balance at a very even keel. It turns out that those hormones are related to each other. They are the product of an ancient implication about 450 million years ago. So initially there was one gene, one hormone, one receptor. There was a duplication 450 million years ago that produced two identical ones. And then those changed by a series of small mutations to give them the specificity they have now. The Thornton lab has worked out that change in detail. They know step by step all the amino acids involved in producing these hormones. Okay, so what do I want, the final message I want to get across to you is this: nature's not an engineer. It doesn't do things by intent. Design is kind of an inappropriate word to use for natural processes. That what's going on is as I say here, evolved organisms exhibit complexity, and they do so because of a bunch of natural processes, which I haven't gone into in great detail. Okay, I'll take my class. Chance, modularity, multifunctionality, incremental tinkering, we talked a little bit about that, and contingency, the effects of history on them. They're all shaped by functional constraints imposed by natural selection, but initially they are produced by simple biology without brains behind them. You don't need brains to build this stuff. You don't need design, you don't need a creator, you don't need an intelligent designer. So, this is the bottom line. Complexity is easily produced by natural processes. I want to touch on one last little subject, though, because I like to generate a little controversy. That one of the messages we want to get across as scientists, and also as many of you are atheists or freethinkers, is that there's a couple of priorities in how we think about the world. That what we want is for people everywhere to use these processes to think about the world. We want them to use reason, evidence, critical thinking, naturalism, and we want them to question everything. So in a moment I'll stop talking and let you ask me questions, you see. You can argue with me. This is the way we like to deal with the world. In contrast, there are many people who believe in these things. Gods and demons, prayers, heaven, hell, all this kind of stuff. You cannot believe in these things if you apply critical thinking, if you demand evidence, if you demand reason, if you use naturalistic processes, if you even dare to question them. Those things all fall apart.